In 1935, a young Australian tourist picked up a strikingly beautiful seashell, and 5 hours later, he was dead.
Mhm.
About that time, the local tourism industry in Australia was booming, and the Great Barrier Reef had become a really popular tourist spot. And so these small ferries would take people to islands where they spent the day frolicking in the shallow waters, fishing, and marveling at the colorful flora and fauna of the reef. In the month of June, a 27-year-old man, Charles Garbut from Queensland, came to the reef with his friends to a lovely spot where the water ebbs quickly. And when this happens, you can suddenly see shells of all shapes and sizes and bright colors. There, Charles found a beautiful shell that may have looked like the one in my hand right now, a shell with a very pretty brown and white pattern. He held it in his palm and scraped it with a knife to clean the shell and appreciate its beautiful patterns.
But as he was doing that, a snail's venomous barb from within the shell pricked his hand. Almost immediately, his hand went numb. 10 minutes later, his lips were stiff. After 20 minutes, his vision blurred. And by 30 minutes, he was paralyzed in the legs. Within an hour, he lost consciousness, slipped into a deep coma. And as you said, about 5 hours later, Charles Garbut, this 27-year-old fit footballer, had died from the sting of a cone snail.
Welcome to the Discovery Dialogues podcast, where we take a deep dive into the history of groundbreaking discoveries in the life sciences. I'm your host, Ani Deshpande. Each episode, we will uncover some incredible, insightful, and often just insane stories behind breakthroughs in medicine. Join us as we journey through the twists and turns of scientific history, one breakthrough at a time.
I'm your host, Pam Itkin-Ansari, a diabetes and pancreatic cancer scientist at the Sanford Burnham Previs Institute in California.
And I'm Ani Deshpande, a cancer researcher also at Sanford Burnham. This series isn't just about the technical details of diabetes, it's about the wonder of discovery itself and the extraordinary paths that led to groundbreaking inventions and the remarkable people behind them, with diabetes serving as our lens to tell these inspiring human stories.
Exactly.
If you are enjoying this podcast, please like and subscribe to our channel because it really helps us spread the word. Our goal is to share how scientific advances have revolutionized treatments of major human diseases, starting with the first season on diabetes.
Welcome to episode 3 of the Discovery Dialogues podcast.
In this episode, we meet animals with the power to kill us and others with the power to heal us. And sometimes they are the same animal.
The snail that caused poor Charles Garbut to die a swift and excruciating death was a cone snail. I'm sure some of you have seen these shells that are shaped like little ice cream cones and sometimes patterned with zigzags or dots or spirals. They even look really harmless and inviting, in fact, and I have a collection of them.
Yes, but these beautiful and harmless looking shells sometimes hide some of the world's most sophisticated hunters, cone snails. In fact, these cone snails are extremely devious creatures. They hunt worms, they hunt other cone snails, and they even hunt fish. And bizarrely, cone snails can swallow an entire live fish swimming nearby on the ocean floor.
But if we think about it, fish are fast swimmers, right? And cone snails are, well, snails.
Yeah, and that brings to mind one of my favorite topics, as you know, Pam, venoms. Cone snails have a unique superpower. They literally have a chemical weapons factory. Cone snails carry a powerful arsenal of venoms that can cause all sorts of damage, from neurological to metabolic to muscular to vascular disruption, and all of this with ruthless precision. They even make tiny proteins that destroy cell membranes. So these venoms have earned the snails, like the textile cone snail, whose shell I have in my hand here, or the one that likely killed Charles Garbut, the nickname cigarette snails, because the victim may only have enough time to smoke a single cigarette before dying a miserable death. Now imagine this, a cone snail lurking on the ocean floor and a fish is swimming by. Suddenly, the cone snail releases a mysterious venomous substance into the water, which makes the fish dizzy and disoriented, and then the cone snail starts sucking the live paralyzed fish directly into its mouth as if slurping ramen from a noodle bowl.
Yes, and if the stunned fish tries to escape, the cone snail has another trick up its sleeve. It shoots out a harpoon-like object with backward-facing barbs like a fishhook, and it plunges that into the fish, delivering a fast-acting substance. Then the fish suddenly slips into a coma, and the slow but incredibly devious cone snail finishes its meal.
Fish hunting snails are particularly dangerous, with venom strong enough to kill a human. This harpoon is cocked and loaded. Target is within range. The cone snail strikes. The fish is paralyzed within seconds, reeled back in, and swallowed.
But Ani, with all this discussion about venoms, this is a diabetes podcast. We are going to get back to that, right?
I promise you, this episode is still on diabetes, believe it or not, although we will have to wait for a little bit.
Okay.
First, let's recount some insane stories of how studying venoms has led to blockbuster medications. Here, a really cool story comes to mind. Up until the 1960s, scientists debated about whether having high blood pressure was a good thing or a bad thing. In fact, the consensus was that lowering blood pressure in patients with high levels was a stupid and even dangerous idea because they thought that lowering the blood pressure may limit flow to vital organs. But some scientists suspected that high blood pressure caused all sorts of problems and was possibly leading to strokes and heart attacks and premature deaths. A pharmacologist, Dr. John Wayne in London, really wanted to test what would happen if you actually use a drug to lower blood pressure.
There was one problem, though. No drug like that existed.
Well, it didn't until Sergio Ferreira, a Brazilian postdoc who had just arrived in John Wayne's lab, told him about a highly venomous snake, the Brazilian pit viper. He told Wayne that when farmers working in cornfields are bitten by this viper, they would cause a dangerous and often fatal drop in blood pressure. Ferreira got some of this venom to London, and this venom, which is a potent weapon from the deadly Brazilian viper, became the basis of one of the most important life-saving drugs in modern medicine, Captopril. Here's one of the most famous venomologists in the world, Brian Fry, talking to Ann Jones from ABC.
If you know of anybody, for example, who takes high blood pressure medication, odds are they're taking Captopril or one of its derivatives. Well, that's a medicine that was developed 40 years ago from a modified snake toxin. It has been one of the top 20 drugs of all time. Its medical importance cannot be overstated. It is up there with aspirin.
This became the basis of most of the anti-hypertension drugs. If you ask anybody you know, like your older family member who takes even the more modern blood pressure medications, they are inspired by the venom of this Brazilian pit viper.
Right.
Pam, when I grew up in India, my own childhood had quite a few real life snake encounters. And that's because I grew up in a city which was just carved out of dense vegetation near the hills outside of Mumbai. So snake sightings for me growing up were not very uncommon. Of course, most of these snakes were harmless, but I have seen a cobra near my school. I actually also remember seeing a Russell's viper when I was in college. And now when I've moved to San Diego, I see rattlesnakes pretty regularly in my neighborhood and once even in my backyard. It's rattlesnake season right now, and I can guarantee you that plenty of slithering snakes in the canyon just behind where we are doing this recording.
When a rattlesnake bites its prey, it releases a cocktail of venomous compounds. But one of the effects that's really horrible is internal bleeding, which can be fatal. So this is a good time to introduce a very important concept, first set out by the 16th century Swiss scientist Paracelsus. He laid out perhaps one of the most important concepts of toxicology, and perhaps even of medicine itself. He said, all things are poison, and nothing is without poison. The dose is what makes poison. The dictum revolutionized modern medicine because it changed how we thought about toxins, and it introduced the idea that toxicity is not an absolute property of a substance, but rather depends on how much of it is used. It laid the foundation for the idea that many highly potent compounds, including venoms, can be turned into life-saving medicines. And so when scientists were looking for drugs that could rapidly prevent blood clots, they couldn't help but notice that rattlesnakes had already learned that trick years ago. And so Integrilin is a drug that's inspired from that venom of pygmy rattlesnakes. And since the venom has been designed to prevent blood from clotting, essentially rattlesnake venom has been repurposed as a medicine and is given to patients during a heart attack or during angioplasty surgery to stop blood clots.
Of course, after all this, there's a growing interest in venoms as medicines, not only from snakes, from many other organisms that are being tapped into for their enormous potential. And the tiny cone snails that we were talking about, they are the source of one of nature's largest factory of potent venoms. In fact, they make hundreds of different kinds of venoms. And one of the scientists who has made seminal discoveries in studying these mysterious creatures is Dr. Toto Oliveira. He's a scientist who spent his childhood in the Philippines, where he was fascinated by cone snails.
Yeah.
So I grew up in the Philippines. I was an only child, and my parents moved to a home that was very isolated. I began collecting shells maybe when I was about 9 years old. That's how I became quite seriously involved in shells.
Amazingly, a student straight out of high school who joined his lab found that one of the venoms in the cone snail was a potent painkiller.
Our hope was that we would have some basic science impact. It wasn't so clear that that would be attained, and this was completely unexpected.
We didn't start out with the idea of we're going to research these cone snails and we're going to come up with a pain medication. But as we got into it, there was this incredible gold mine of compounds.
And this led to development of a potent analgesic, ziconotide, that was approved by the FDA in 2004 for use in humans. And you know, Ani, generally, when we try to take a venomous substance to make a drug, it has to be modified by chemists. But one of the things I couldn't believe about ziconotide, it is the same molecule made by the cone snail that we use. And in fact, my daughter is an anesthesiologist, and she told me she uses ziconotide when patients are admitted that are in extreme pain. It's amazingly effective.
Now, if you're a predator, why would you spend your energy in making a painkiller to ease the pain of your prey?
I know it sounds counterintuitive. You're trying to hurt your prey.
Well, as it turns out, this is a very common trick in the animal kingdom. Many animals, when they bite their prey, they inject painkillers or even anesthetics so that they can numb their prey before the full effects of the venom kick in. It's like disabling the alarm systems of a building before breaking in.
Oh, exactly. Okay, Ani, I think we're having so much fun with venoms. We've made the audience wait long enough. And we should tell them what these fascinating stories have to do with diabetes.
Yes, I think we should.
Okay, let's come back to the venom of this cone snail. We were so fascinated by these creatures, we decided to reach out to Dr. Helena Safavi at the University of Utah, a scientist who made an astonishing discovery about the venom. On a Wednesday afternoon recently, we met Dr. Safavi on a video call. She had a relaxed and approachable presence and a friendly smile.
I think she had the vibe of someone who might tell you a wild story about killer snails over coffee
Right.
and then just go on to mention that she's trying to turn these dangerous venoms into medicines.
Exactly. Yes, born in Iran, Dr. Safavi grew up in Germany where she learned to scuba dive in lakes. But she soon realized she wanted to study wildlife and was especially interested in the ocean-dwelling creatures. And for that, she needed to move. Here's Dr. Safavi.
I think I always loved being in nature and just the natural world. I always found very fascinating. I also really love animals, like all kinds of creatures I always really liked. So even as a young kid, I would like to be outside and had this admiration for the natural world. I didn't know that scientist was a job, so it was just like a natural curiosity. And then in addition to that, I grew up in a very small town in Germany, and there wasn't that much going on. And we were far away from the ocean, far away from any like really thriving ecosystems. So I had this fascination with like different places that would be full of life and biodiversity.
That's
Great.
interesting. I also spent a lot of time in Germany. I did my PhD and postdoc there.
Yeah, so there's very little wilderness left. So I think I just had this dream of going to a world that is so different to our own.
Oh. And why she was interested in venoms?
I realized I did my PhD in Australia, and during that time, or actually even before my PhD, I realized that I wanted to study cone snails. And Dr. Oliver is really the world expert in cone snails, so he knows everything there is to know about these creatures. There are so many venomous animals out in the world that are just fascinating.
And unlike what goes on in also good, but in Dr. Safavi's kind of work, I guess they get to travel to exotic places.
Oh.
My graduate students, I shouldn't tell them that your students probably get to go to the Solomon.
Don't think I took any students.
And they get to go to Boston or Chicago, right?
You have to be pretty dedicated to make it on one of those trips. So.
Yeah.
Cone snails can be found everywhere, but some of the most interesting ones are in the tropical oceans of the Indo-Pacific region. I'm sure you have fun stories, Dr. Safavi.
Yeah. The things that I can tell.
Right.
Yeah, so there's one fun story. So when we go out to collect these cone snails, we go during the day, but most of the time they're active at night. So they're nocturnal animals, most of them. So we go out at night to go collecting, and the night always has a lot of surprises for you. So we go diving, and this was in the Solomon Islands. And then they still, if it's not like super dark yet, they hide in the sand. And so you normally go in and you dive, you go above the sand and you start to, try to get the sand or disturb the sand so you can find the cone snails. So we were disturbing the sand, and all of a sudden we open this like field of stingrays under us. And there were probably like 20 stingrays right under us, and we're like we're disturbing them. And we got so terrified.
Yeah.
And so ever since then, I go down, I disturb the sand just a little, just to make sure that there are no stingrays hiding, and then we continue.
One night when there was a thunderstorm, Dr. Safavi went to collect exotic cone snails off the coast of the Philippines.
We went during a thunderstorm, and I actually still don't understand I don't think you should be on a boat, a small fishing boat, and that was in the Philippines during a thunderstorm. So there's this thunderstorm, and I go and ask, is it safe to go diving under these conditions? And they say, we do this all the time. It's totally fine.
The locals?
The locals. And it's true, they do that all the time. So then we go under, and it's the most amazing reef at night that I have ever seen. Everything was so active, and I think it was because of the lightning and the thunder. Like all the animals on the reef were rushing around and being busy, so that was just really wonderful. So ever since, I've been waiting for a thunderstorm so that I can go scuba diving again.
And they captured the cone snail, the Conus geographus, the one that swallows its fish alive, and were so captivated by what the snail can do because it paralyzes the fish by sending a spray of venom into ocean water before devouring it. And she wondered, what kind of substance could do that? She had to know. And what she discovered was astonishing. The venom that this cone snail was making was insulin.
Here's ABC4 talking about Dr. Helena Safavi's work.
Now, in other news, there is fascinating research being done right now on snails at U of U Health. Now, did you know there are thousands of snails that use poison to capture its prey? That venom includes insulin, and researchers believe it could help people with diabetes.
We had never seen insulin in any animal venom ever, so it had never been described, and we were extremely surprised to find an insulin in these snails.
And just like in humans, the hormone insulin regulates blood sugar levels, but for these predators, it reduces insulin in their prey almost instantly. Dr. Safavi believes this brings them closer to developing faster-acting insulin to treat diabetes in humans.
And now we started to use the insulin that we found in the cone snail to see if we can make better drugs for people that suffer from diabetes. And the fact that we can explore it to improve human health makes us feel very fortunate about the work we do.
So let's just pause for a moment. What the cone snail was doing is that it was spraying insulin into the water so that the fish went into this hypoglycemic state.
Yeah.
And if that wasn't enough, it also injected the fish with insulin through its harpoon-like needle. So in a sense, it was weaponizing insulin for murder.
Yeah. Well, yes, actually for hunting, but you're right.
Pam, this is so interesting, and this weaponized insulin
Yeah.
actually reminds me of a rather dark episode in the 1990s, and this is a real life episode. In a hospital in New Jersey, they noticed that many of their patients,
Yeah.
their blood sugar would just suddenly and mysteriously crash.
Right.
And so they would need emergency medication and sugar. And oddly, some patients on IV drips would suddenly see an improvement in their condition as soon as their drips were taken off. And so a careful investigation revealed that there were extra puncture marks on their IV bags that has saline or any other medications. And so they found that someone was actually injecting insulin into these IV bags. And this was an ICU nurse. His name was Charles Cullen, and he was later convicted for the murder of 29 patients, although it could have been many more, even in the hundreds.
Yeah.
This story was actually dramatized in a very popular Netflix series called The Good Nurse.
Oh.
Here's a short clip from the Netflix trailer.
Nurse Lochran, this is Officer Braun.
Do you remember Anna Martinez?
Yeah, her death. It was sudden.
Mind taking a look at this?
Oh my God. There's insulin in her system.
And it's absolutely insane and shows how insulin is a double-edged sword. In the last 2 episodes, we talked about how life-saving insulin is, but as anyone who uses insulin for diabetes treatments knows, too much insulin can be life-threatening. And it's really wild to know that in the animal kingdom, some animals use it as a weapon for hunting. We asked Dr. Safavi how she figured that out.
I did a short internship while I was a student at the University of Cologne working on insulin and the insulin receptor. And I had looked at just the insulin sequence for that a few times. And so when we identified the compound in venom, and I just looked at the sequence, it was obvious then that that was indeed an insulin. And people had looked at the sequence before. So the species that we found the first insulin in is called Conus geographus. It's a very well-studied cone snail species. Many people have looked at its venom, and people kind of overlooked the fact that it's an actual insulin, because you have to kind of know what you're looking at. So I think that previous experience definitely helped with that discovery.
Excellent. Louis Pasteur has said that chance favors the prepared mind. And there are so many examples of this in science. And we wondered, how does insulin released in the water drop the blood sugar of fish? It can't be just by going in the mouth, because as any diabetic patient knows, insulin taken by mouth doesn't work, which is why it has to be injected into the body. So what's going on here? Here's Dr. Safavi.
And can you explain the difference between the cone snail using a spear to inject venom versus releasing it into the water? Because I'm wondering if it's released into the water, how does it even get into the fish? Is it through gills or is it ingested?
Yeah. So most cone snails that we know of and that we have studied use the injection method. So they directly inject the venom into the prey. There are currently only 2 species of cone snails that have been observed to use this so-called net hunting strategy, where they open like a large fake mouth and they start releasing venom into the water. And the venom cocktail can get into the fish's body through the gills. So that was right. Some of it may go through the mouth, but it might not be that bioactive. But when it goes through the gills, it enters the bloodstream pretty quickly. And that's how it can act quite effectively in the fish. So when we first discovered this insulin, we knew it was from this species that releases it into the water. And I started to look around, and there's actually a very nice lab experiment that people do to learn the effect of glucose on the body, which is you take a goldfish and then you add human insulin into the water, and the goldfish fairly rapidly stops swimming and becomes hypoglycemic.
So the fish are getting insulin into their bloodstream through the gills, which are their respiratory organs, and that's why their sugar drops dramatically and they become unconscious and actually float to the surface. But if you're quick enough,
even human insulin in the fish can get through the gills. And then you can take the fish and transfer the fish to a glucose bath. So now the water is supplemented with glucose, and then it very quickly recovers.
Oh, this is just like my relative who keeps glucose tablets on hand and quickly pops one as soon as he feels a sugar low coming on.
Yeah, and I've had people work in my lab because they are diabetic and they want to work on a cure, and they have to keep sugar tablets with them too. And if the cone snail insulin can get in through the gills, it makes you wonder, can we ever make an insulin for diabetics that could be inhaled through our lungs?
People are trying, or we're trying to develop inhalable insulin.
Yes.
So there are ways to get insulin to cross through epithelial layers, such as those in the lungs and also in the gills.
But why does cone snail insulin work so well in the fish? Dr. Safavi gave us a shocking insight. The cone snails make 2 kinds of insulin, one for their own metabolism, like most other organisms on Earth, and the venomous insulin that's used specifically for hunting fish.
That's really crazy. But Pam, I don't eat fish, and I'm sure some cone snails are not fond of fish either. So what do they do?
Well, honey, I'm not sure I follow the logic that if you don't eat fish, some ocean-dwelling animals would change their diets. But in fact, you're absolutely right. Even though Conus geographus, the fish hunting cone snail, makes fish like insulin, there are cone snails that hunt either other cone snails or even worms. And as Dr. Safavi explains,
but there are so many species of cone snails. There are around 1,000 different species, and many of those hunt other snails. And when you look at the insulins in those species, those insulins look like snail insulin. So although we've never tested those, we're pretty sure that they're also being used to hunt other snails, or to hunt snails in that particular case.
Interesting.
This is so fascinating. I'm just gonna summarize this so that I understand this. What you're saying is that some snails, like Geographus that you studied, make insulin that is actually not snail insulin, but they make an insulin that is more similar to the fish and used mostly for fish hunting. But then you say that there are probably other snails that make insulins that are similar to snail insulins because
they're going to hunt.
They're going to hunt
those,
those,
or others hunt other snails.
And we know that those snail hunters make those snail insulins, and to add another layer to it, there are species that hunt worms, and those make insulins that look like worm insulins. So those snails are really
I love
amazing.
So a question that comes to mind, of course, is now that the cone snails have all these weapons, how does it protect itself from the highly potent venom?
Well, there are various ways, and we will come to cone snails in a moment. But generally, venomous predators do a lot of different things. The first is behavioral caution. Predators usually don't bite themselves.
Yeah, that's true.
But there are more interesting ways. For example, some venomous receptors so that the venoms don't affect them, or they target processes that are only active in their prey but not in their own
body.
Or, interestingly, sometimes they make venoms that become active only after the injection into the body
yeah.
of the prey. Sometimes venomous predators also make proteins that will bind to the venoms and keep them in an inactive state.
Like a proto-venom then.
And then there is another way, which is called compartmentalization, which means that the venoms are just stored safely away from the rest of the body.
It's actually true for all the toxins that are in the cone snails. And the way the venom apparatus is located in the body, it's compartmentalized. So it's very separate from all the other organs.
Yeah, so the venom sac solves two problems. One is protecting the cone snail from itself. And two, it provides a separate depot of concentrated insulin that's ready ammunition for deployment whenever it's needed.
I find that amazing.
Wait, we're not done yet. Remember when diabetic patients inject themselves with insulin, they tend to do it with a new needle each time. Otherwise, it would become very blunt, like in the old days. So what do cone snails do about the harpoon? Does it get dull?
And when the cone snail is done with its meal, it'll actually regurgitate the bones, and you will see that little harpoon, and sometimes multiple harpoons come out with the bones. So this is how we know that in addition to releasing venom, there are additional injections of venom into the
So
fish.
they just get rid, like teeth, like animals, make new teeth.
Yeah.
Right?
Yeah.
Yeah.
So I was discussing this with my son, who is 15 years old, and he said that they probably make more harpoons.
Yeah, they can constantly remake them. There is what is called a regular sac, which is an organ that is attached to the venom apparatus, and that has many ready-to-go regular stored
So it has
in
a quiver.
Yeah, exactly. Yeah,
Full of arrows. That's so great.
But how does this story relate to humans? Remarkably, Dr. Safavi found that insulin from cone snails works on human cells. This is super exciting because understanding the cone snail insulin may lead to novel therapies. As life-saving as human insulin is, there are aspects of it that are not ideal.
I had a question because I've been studying therapies for diabetes for so long. Is there something about the insulins in these venoms that you see that could particularly be useful compared to human insulin in terms of stability, temperature sensitivity? For instance, right now, human insulin has to be kept cold, and that's a big problem in developing countries, right? Where they might not have refrigeration. Do you see a way right now toward a simpler insulin that's more stable, might not need cooling, that sort of thing?
Yeah, so on that, we have done some work on making a fast-acting monomeric insulin based on the venom insulin. So that doesn't necessarily immediately address the stability issue, but human insulin is basically a sticky molecule. One insulin likes to stick to another, and they form this hexamer, it's called, basically many insulin molecules together.
Yeah.
Right.
And when you inject that, it takes some time for the insulin to act.
Because it has to come apart from the zinc for it to be active, right?
Exactly, and it can get stuck in the tissue. Yeah, and so when we discovered the venom insulin, it was clear that all the venom insulins that we have studied to date are monomeric.
So to clarify, human insulin's released from our pancreas as 6 molecules held together. And when it gets into the bloodstream, it comes apart into individual monomers that are bioactive very quickly. But when human insulin is packaged for patients, it is stable, stored as a hexamer. That is the 6 together. But because it's injected under the skin, it doesn't get into the bloodstream right away, and it has to dissolve into that monomeric active form. Cone snails just make a single monomer form, and it acts very rapidly. If we can make a monomeric form that works in humans, it might be faster acting and potentially shelf stable. Ani, we've been talking about insulin in all our episodes and how it's absolutely essential for blood sugar control. But it's also critical that our bodies have a way to compensate for low blood sugar. The way it works is the pancreas senses the danger and quickly releases another hormone into the bloodstream. This hormone is called glucagon.
It's easy to remember this name because it's literally derived from the Greek roots gluk for glucose and agon for agonist or mobilizer. So glucagon is the hormone that signals to the liver to release stored sugar when you're fasting or during periods of low sugar.
In fact, some patients keep an injection of glucagon handy so that if they feel themselves going into a state of low blood sugar, they take an injection of glucagon to speed the release of stored sugars.
So when the fish is attacked by the cone snail insulin, why doesn't it just counterbalance it by releasing glucagon?
One very powerful inhibitor of glucagon secretion is somatostatin. So the cone snail in this cocktail of toxins has an insulin to first induce low blood glucose, and then it has the toxin, the somatostatin-like toxin, to inhibit the glucagon secretion. And that has the effect that the fish cannot easily recover from the low blood glucose, but can be caught more easily.
Right, that they're really stunned for long enough.
It's
for
a
the
1-2 punch.
Yeah,
Right.
punch.
And we know that there are many other toxins, and we think that there are additional ones that act on glucose homeostasis, and we're currently investigating those in the lab.
Oh my
too.
gosh.
Okay, so what's going on here is the cone snail not only makes insulin to drop the blood sugar, but it also makes another fish hormone to prevent the fish from recovering. And this arsenal of chemical weaponry from cone snails could one day lead to many new potent medications for diabetes. Just imagine, too, there are many venoms out there that could regulate blood sugar in ways we have yet to find out.
Absolutely. And one of the reasons we made this episode about venoms is that in a later episode, we will narrate the incredible story of how the venom of a completely different and very unlikely animal from the Arizona desert has already led to one of the most game-changing diabetes medications ever.
These are amazing stories, Ani, but you might be surprised to hear how incredibly sad they make me, because they emphasize the danger of an insulin overdose. And as a diabetes scientist, I've heard from many patients, and especially parents of patients, how much the fear of accidentally taking too much insulin impacts their lives. If you remember, high blood glucose leads to several complications over the long term, but low blood glucose is immediately dangerous. So patients walk a tightrope, trying not to go high, but very afraid of going low. In fact, what they're afraid of has an ominous name. It's called the dead in bed syndrome, which refers to the sudden unexplained death of a person with type 1 diabetes while they're asleep. Thankfully, this is very rare, but still, just the thought of it is enough to keep people worried and anxious. Parents have told me that they have never slept through the night since their child was diagnosed with diabetes. They set an alarm to check the blood sugar in the middle of the night.
In future episodes, we will talk about the many exciting advances that are helping patients deal with low blood sugar.
But we have to end this episode on a happier note. And I know you have a really fun story to share.
Don't I always? We leave you with the story of Lelia Bullion. When she was just 8 years old, she had what the family suspected was flu, but her mom, who was a nurse, had her doubts. Lelia had symptoms including excessive thirst and weight loss, and when they checked her sugar at emergency care, it was 700, which is really high. The normal range after a meal is less than 140. Here's Lelia sharing her story with University of Colorado science writer Deborah Melani in 2022.
We went to the Barbara Davis Center in Aurora and spent a long, 10-hour day there just learning all the ins and outs of diabetes, and I remember the feeling. I was terrified because I was just like, what is this? An 8-year-old being diagnosed with something that I've never heard of before. 2 years into having diabetes, I've had multiple seizures before. So oftentimes those were nights where I had heavy practice and I just didn't eat enough and I thought my blood sugar was okay. going to sleep was troubling because we were worried that I would have a low in the middle of the night.
And that day at the emergency room, she got a shot of insulin, which gave her a temporary reprieve. But this 8-year-old and her family now had to think about ensuring that her blood sugar was okay every day for the rest of her life. For one thing, this meant getting her finger pricked 10 times a day with a needle. And here's Lelia's dad.
When she did have lows, we would treat her, but yet we would stay up with her for hours. Or we would sit in bed with her or sleep with her and check her again. And as a parent, I love my kids more than anything in itself, and not knowing what we would wake up to.
Her family then turned to local heroes. Kiwanis is an international organization of more than 600,000 volunteers dedicated to improving the lives of children. It turned out that in Lelia's own state of Colorado, one chapter, Foothill Kiwanis of Boulder Valley, specializes in alert dogs. We spoke with Alan Bove, the program lead and trainer. They provide diabetic children with a free puppy that the child and their family then trains with volunteers from the Kiwanis Club. Buying an already trained alert dog is very expensive. The Kiwanis chapter gifted Lelia an 8-week-old golden retriever named Gizmo. And over the next 2 years, the Kiwanis program taught Lelia and her family how to train Gizmo to be a living and breathing continuous glucose monitor.
Now, it doesn't give me readings of my actual blood sugar, but Gizmo alerts me before my blood sugar is low so I can catch it before anything happens. In the middle of the night, if he smelled me, I'm a heavy sleeper, so oftentimes he couldn't wake me up, but he would go out of my room to go get my parents and bring them to me.
He will jump on the bed, and I'll feel it immediately, or he'll put his head under the covers and grab my arm and pull me, so I immediately know something's wrong and get up immediately and go into the bedroom and check on her.
He gave us an incredible peace of mind. Gizmo has a certain command where if I get too low, I get really shaky and my legs just feel really weak and I can't comprehend things very well. So at that point I have Gizmo right next to me and I tell him, I say go get help, and he'll bark profusely and just get somebody's attention. And as soon as I say go, like go get help, he'll go find the nearest person, whether it's my brother, my mom, my dad, anybody's in the house, a friend, to come and attend to me because I can't do it myself. 3 years in, I was pretty consistent, pretty independent with my care, so I would highly recommend service dogs. Like, they're incredible and they're super accurate and they're great companions to have, and they're fun to bring around and walk around with.
We caught up with Lelia for an update this week, and she is now in college and Gizmo is still by her side.
Even though I'm back on the CGMs now, technology is still improving, but if I didn't have him, I think I would still be like a little scared of diabetes.
What was really amazing is that sometimes Gizmo could detect a sugar low before the monitor could.
My monitor will say, I'm at like 120 and steady arrows, nothing going up, nothing going down. But Gizmo's like alerting to me. He's like, something's wrong. And then 15 minutes later is when I start seeing the trend that I'm going low.
Oh, I love that. Over time, the relationship between Lelia and Gizmo grew, and he was more than a medical alert dog. He was a source of comfort, courage, and companionship. Gizmo walked with Lelia at her high school graduation and now accompanies her to college, where she's training to become a pediatric nurse, a role inspired by her own journey with diabetes. Today, with Gizmo by her side, Lelia lives a life of confidence. She coaches young gymnasts, and she dreams of comforting kids who, like her, once felt scared and uncertain. Lelia's story is a powerful reminder. Even with a chronic condition like diabetes, you can live a fulfilling life, you can dream boldly, and inspire others, especially when you have the right tools, a good team, and a furry friend like Gizmo.